I am live. Good. Okay. Well, I guess that's why we test things. Okay. So, who thinks they can answer this? All right, let's go here.
The natural killer cells kill cells that are infected with intracellular parasites. Or, sorry, not parasites, pathogens.
Like
Viruses.
Like viruses. There's another type of cell they kill. Do you know what it is? Yes, cancers. Good. So, which ones do they kill? Do they kill all viral infected cells and cancers?
The ones that present abnormal or don't present.
What does abnormal mean in this sense?
Non-self.
So you mean that they're presenting non-self antigens?
Yes, associated with the MHC1.
Not quite true. Those are killed by a different type of cell. Those are killed by cytotoxic T cells. Natural killer cells kill what I think you were just about to talk about. So ones that present abnormally, ones that present non-self antigens are going to be killed by cytotoxic T cells. What was the other thing you were going to say? Or that don't?
That don't present any antigens.
That, yes. Those are the ones that are killed by natural killer cells. Natural killer cells kill cells that that don't present antigens on an MHC1. We'll talk about MHCs today, but basically, you remember I said at the beginning of this section that your cells have to sort of show their papers. They have to show a display of whatever proteins they are making at that time. And one of the things that can get around destruction by cytotoxic T cells is if you just simply don't show anything. Cytotoxic T cells can only kill you if you show abnormal proteins. What if you just don't show any proteins? Then cytotoxic T cells can't do their thing. But natural killer cells, their job, and the reason why they're natural killers, is that they're non-specific. They kill any cell that is refusing to show what proteins it makes. Natural killer cells also do a few other things. That's their main job. But natural killer cells can also be leveraged by antibodies to kill any cell labeled by the antibodies. This is something that I'll talk about in a video that I'll upload probably tomorrow on antibodies and how they work. But in this case, it's called, I believe, ADCC, antibody-dependent cellular cytotoxicity. And so in this case, cells that are labeled with some types of antibodies, not all types of antibodies. It's mostly IgG, which I'll also explain the different types of antibodies later on. But cells that get labeled with these types of antibodies, natural killer cells, will get up to them, will recognize the antibodies on them as saying that you should destroy this cell, and then they release their death package. The death package, which, by the way, is a pretty cool name, basically consists of a whole bunch of nasty enzymes, toxic chemicals, things that will stimulate apoptosis. It's a death package. You can guess what it does. Anyone have any questions about natural killer cells and their function in the immune system? All right. Then I'm going to end that and create a discussion forum here. All right. Adjustments only, save, and go ahead and present this. All right, I'm going to try to remember to refer back to this more frequently than normal because that's part of the whole testing out this whole attend from home thing. Everyone got the announcement earlier today, right? I put an announcement on Canvas. Not that we are, but that we are preparing to do so. Like, they want us to be prepared in the eventuality. And also, I'm like asking people who are sick to please stay home. And like, that makes sense. If you're sick, you should stay home. But I don't want people to think, oh no, I got to make it into class because I got to get that information. Which you do. So in order to encourage people when you're sick to stay home, I'm having an attend from home option where I'm going to like stream the lecture. And if you go through Top Hat, you should usually, not today, you should usually get the slides displayed at you. And then I'm going to stream my voice and you can type in questions into the questions, ask them here forum.
Are you still in school?
Yes. Class is still being held. I'm just putting an option out there for students who may be sick or have a loved one who is sick that they have to take care of. All right. Our topic today is B and T lymphocytes and the acquired immune system. So we talked a little bit about B and T lymphocytes early on, and I noted that the B lymphocytes are primarily involved in the humoral response. Cytotoxic T lymphocytes are primarily involved in the cell-mediated response, and helper T lymphocytes are kind of involved in both. Now we're going to put some details on that. And actually, in order to tell you guys the story of the B and T lymphocytes and acquired immunity, the first thing that I have to do is actually go to something that you know of already. Antigen-presenting cells. Who can tell me what the antigen-presenting cells are? It's okay, I don't have a volunteer. I can call on people.
All cells.
All cells? No, not all cells are antigen-presenting cells. The antigens that we're talking about here are foreign antigens, if that's any help. Say it again. No, those are ones that display self-antigens. We're talking about ones that display foreign antigens. Yes. Phagocytic cells? Close. Not all phagocytic cells, but many of them. Yes?
Dendritic cells and
Dendritic cells, macrophages, and monocytes, the mononuclear phagocytes. Are there other phagocytic cells?
Yes.
Name one. Neutrophils, yeah. Neutrophils are not antigen-presenting cells. Say it again.
B cells.
We're getting to that. You're a step ahead of me. So, other than the mononuclear phagocytes, dendritic cells, macrophages, and monocytes, there is one other that I will talk about when we get to it, because it's a little special and weird. But these are antigen-presenting cells. I told you guys a little bit about them when we talked about the cells of the immune system. So here we have a dendritic cell. It's hanging out. It's a dendritic cell, so it's probably hanging out near some of your barriers. Here we have a bacteria that I probably should not have done in dark red on a black background, but hopefully you can kind of see it. The dendritic cell does what dendritic cells do. It's going to detect that bacteria through using its, what type of receptors? Say it again. Nope, not yet. These are pattern recognition receptors. They're a type of pattern recognition receptor that we talked about. Toll-like receptors, yes. Remember, toll-like receptors are going to detect common pathogen motifs, what are called PAMPs, or pathogen-associated molecular patterns. Things like flagella, fimbria, peptide glycan, pili, things like that. Right? Things you commonly find on bacteria, do not find on your cells. So it's recognized this as being a bad one, and it has eaten it. And of course, when it eats it, it takes it inside into a phagosome. And then the phagosome combines with the lysosome and to make a phagolysosome, which is going to chop up that bacteria into little itty bitty 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 pieces. By the way, it doesn't have to be a bacteria. It could be a virus. I was using a bacteria for now. Now, it doesn't stop here. If it was a neutrophil, this thing would now just go blah and split out all of the dead pieces of bacteria. But this is an antigen-presenting cell. So while it's going to expel most of this, it's going to take a selection of all of the antigenic proteins and polysaccharides that it finds. An antigen is anything that generates an antibody response. Antigen. Usually, they're going to be small pieces of proteins or polysaccharides, but they have to be like unique. You don't usually find lipids making good antigens because most lipids are very similar. So anything that binds to a lipid is going to bind to lots of lipids, and you want antibodies to be as specific as possible. So a whole selection of antigens is going to get presented by this dendritic cell on its surface, on an MHC2, which is called a major histocompatibility complex class 2, or we're just going to call it MHC2 from now on. MHC2s display foreign antigens. They're found on cells that eat foreign things, and they display those foreign things on them, not self-antigens. So this dendritic cell has eaten a thing and is now displaying an antigen from that thing. It would actually be displaying a whole bunch of different antigens, each on a different MHC2, but I'm only going to show one because I'm lazy. And now it's not going to do any good hanging out like in your skin or whatever, right? It's got to go where it can show something who knows what to do with that information. That means lymphocytes. It needs to show this antigen to lymphocytes. So it's going to go where the lymphocytes are. And lymphocytes can be found all over your body, but they're found at very, very high concentrations in lymph nodes and in other lymphatic organs. So your antigen-presenting cell is going to go to one of these. We'll just say it's a lymph node. It usually is. It's going to go to a local lymph node, whatever's closest, and it's going to hang out in here. And here's where we're going to pause the story of the antigen-presenting cell, because what we've got is a big narrative here. There's a lot of different characters and a lot of different interactions. So this guy's story is to be continued. And now we're going to take up on a different character's storyline, the B cell. First off, what is a B cell? Well, B cells are lymphocytes. Like all lymphocytes, they are born in the bone marrow, but they are also trained in the bone marrow, which not all lymphocytes are. So I want you guys to think B cell, B for bone marrow. Right? And when I say they're trained, what do I mean? Okay, so every B cell makes B cell receptors. Here you can see a B cell receptor here. B cell receptors are almost exactly the same as antibodies, except they're stuck in the membrane instead of floating around free. Every B cell is going to have a different B cell receptor. Some of them are similar, or actually, sorry, back that up a little bit. All of them have similar parts. So for instance, they're all shaped like a Y, and the stalk of the Y is going to be the same in all of them, and like down to about here on the arms of the Y is going to be the same in about all of them. That's called the constant region. But at the ends of the Y, like where my hands are, is the variable region. And every different B cell receptor, which is going to be different on every B cell, is going to have a little bit of a different shaped variable region, which means it's going to grab on to different things. And you could have billions, tens of billions of B cells in your body. And each one of them has a different B cell receptor with a different variable region that binds different things. Now, each of those is a different protein. Proteins are made by genes, or at least encoded for by genes. How many genes do you think you have if you have billions of different B cell receptors?
We have less than the amount of B cell receptors.
Well, yeah, a lot less. Anyone know? Like 25,000, 20 to 25,000, maybe up to 30, depends on your definition of a gene. But specifically, you only really have 2 antibody genes, which are what you're going to use to make B cell receptors. So how do you get tens of billions of different proteins from just a couple of genes? Well, I'm going to explain this in detail in the video I upload about antibodies. But just to sort of give y'all a preview, anyone here been to Chipotle? Most of you probably. Would you say that Chipotle uses a lot of ingredients in their food? Not really. Like, there's a little bar there, and it's like, all right, you want rice? No? Beans, black or pinto? What sort of meat? What sort of salsa? You want some cheese? All right. Should I wrap it in a burrito or give it a bowl or whatever, right? I was sitting in Chipotle one day, and they have like little factoids on their cups. And it said that there were over like 250 million different meals you can order at Chipotle. And I thought, well, that can't be right. But then, you know, you sort of think about it and like, yeah, because you could get white rice, pinto beans, steak, cheese as a taco, or you could do the same thing as a quesadilla, or the same thing as a burrito, or you could do the same thing except no rice and make that a bowl. Or you could do extra rice, make that a bowl. Or I'm kind of hungry for Chipotle now. But you kind of get the idea, right? You can take a very few ingredients and you can combine them in a bunch of different ways. And that's the way these genes work. What they have is they only have a few sections, but like you pick one of 32 things from part A, and then you pick like one of 16 things from part B, and like one of 5 things from part C. And there's different ways that you can glue them together. And, you know, that means that you have this huge amount of variation in the genes that you can make from it. Just like, I used to call this the Chinese menu model, where you do like, you know, okay, so you pick one cut from column A, one from column B, and you make your own combo. You get to make your own combo with these things. So when B cells are being trained, there's basically 2 steps to their training. First, they start off with this big complicated gene, and they've got to decide what B cell receptor they're going to make. So that's where they make their burrito, right? That's where they're going to go, okay, give me this version of part A, give me this version of part B, give me this version of part C, run them together, and I've got my own unique B cell receptor. And then in part 2 of their training, because eventually whatever B cell receptor they have is going to be a potential antibody candidate. Do you want antibodies that label your own body? No, you do not want antibodies that label your own body. That's bad. So what you're going to do is you're going to take your B cells and you're going to run them through this training course where they get exposed to basically all of the external proteins and polysaccharides and all of your self-antigens. Everything that your body makes that would be visible to them. And if they bind any of that, they fail and they die. All right, like, sorry, you bound to the pancreas, you got to die, buddy. And anything that makes it through doesn't bind to any of your antigens and should therefore, theoretically, be safe. So we got our B cells, and they got B cell receptors. And every B cell receptor is going to be unique. And if a B cell becomes activated, then it's going to turn into what's called a plasma cell. And plasma cells make antibodies. Seems simple enough, right? Well, now let's complicate it. So there are 2 methods to making or to activating B cells. Technically they're called the T-independent method and the T-dependent method. I like to call them the 1 key and the 2 keys method. The simpler, the less common one is the T-independent method. And I'm going to start with that. So here's the idea. You got a bunch of B cells. They're floating around in your blood or your lymph or wherever. And note that they each have their own unique B cell receptor. Here's the 1 key method, the T-independent method. So we got this guy come in here. This is a bacteria. Is a flagellated bacteria. It's going to be important in a moment. See this flagella here? It doesn't have to be a flagella, but in order to do T-independent activation, it has to be a special antigen. Not every antigen can do this method of activation. The properties that it needs to have are that you have to have repeated copies of the antibody or of the antigen. So you have many copies of them. They have to be repeated. They have to be close, and they have to be evenly spaced. So for instance, here, maybe this antigen is going to be the flagella, one of the proteins that makes up the flagella. And when you make the flagella, you got the same protein repeated over and over and over again. So an antigen, which is going to be a specific part, one small part of that protein, might have it here, and here, and here, and here, and here, and see how they're like repeated and evenly spaced. Everyone got that? So does this antigen look like it's going to bind this B cell receptor? Like these are kind of squarish. That's kind of circular. Nah, not really a match. So it bumps into that thing. Nothing happens. Bumps over here. That's not really a match. This could take days, by the way. Well, this thing is swimming around your blood, and you got a whole bunch of these things swimming around your blood or swimming around your body or whatever, before they run into the one B cell that has a receptor that matches that antigen. So you got like one B cell in your body that has a receptor that matches the antigen, and when this bugger runs into it, boom, you get a match. But note, it's not just binding to one receptor. It's binding to multiple receptors and gluing them together. These receptors are now going to be stuck in that configuration, and they're brought close together by the repeated, evenly spaced antigens. Everyone with me thus far? Kind of? No? All right. This is a specific type of activation. When you have many receptors activated at the same time and brought close together, that will cause this cell to activate. And what does it do when it activates? Well, it's going to internalize its B cell receptors. It's going to differentiate into a, let's see here. It's going to differentiate into a plasma cell. And when it turns into a plasma cell, it's actually going to grow a lot of cytoplasm, and specifically a lot of endoplasmic reticulum, rough endoplasmic reticulum. What does rough endoplasmic reticulum do? Protein synthesis. And that's because, here we are. One moment. Now let's see whether or not I can. Nope. All right. The protein that it needs to make is antibodies. And it's going to make these antibodies and export them all throughout the body. And oh, and by the way, it's going to reproduce a whole bunch of times because one cell isn't going to make enough antibodies to do anything. So this is the one key method. I call this the one key method because it only requires one thing to activate this cell. All the cell has to do is bind the antigen, and then it activates. This is much faster than normal activation, two-key activation. But it's also more dangerous. So some things that you should know about T-independent activation. Like I said, it requires very special antigens, antigens that are evenly spaced, usually non-protein, and there's a lot of them. These are pretty rare. There aren't a lot. There aren't very many of the antigens around, but there are two specific ones that I want you to know are associated with this, and that is polysaccharide capsules and LPS. There are a few other T-independent antigens as well, but those are the two I'm going to expect you to know on the test.
Polysaccharide capsules.
LPS, lipopolysaccharide. Next thing that you need to know about T-independent antigens is, and this won't necessarily make any sense yet, but it's going to make sense by the time I get done today. There is no class switching, and there is no memory. So that means that if you got infected by this thing and it activated the T-independent antigen pathway and you got better, you haven't acquired any immunity to this. You could get reinfected by it next week. Well, probably a little bit longer than a week. But you know, as soon as like the active antibodies that you made fade away, you don't have any, you don't maintain any memory of it. You haven't acquired anything. Yes.
So then this is kind of like what it kind of just sounds to me like it has like an aspect of it being an innate immunity in that in the sense that it doesn't actually produce memory, but it's using antibodies, which is considered part of the specific.
Yeah, yeah. Remember when I said that things don't necessarily fit into neat, happy categories that we would like them to? That's a good example of one. It does it doesn't acquire you anything, but it does use antibodies, and it uses B cells, which are involved in the innate or in the specific or the acquired immune system. This is specific, but it's not acquired. So yeah, it does have some aspects of both. All right. Any questions about this?
BDK doesn't code. Is that because you're writing your scheme?
It's because I'm loosening my attendance policy. So people will if they're sick be attending from home, and I, you know, whatever, I don't care. Okay, no questions there yet. All right, now let's talk about the other way to activate them, T-dependent activation, or the two-key method. This is why I call it the two-key method. I'm a child of the 80s and grew up during like the end stages of the Cold War. And there was this kind of genre of movies at the time, which, you know, roughly you could call The Russians Are Coming, where, like, the Russians are coming. It's pretty self-descriptive. And we got to launch the nukes and we're going to kill the Russians. And then, like, you know, there's usually some strong-jawed hero or whatever, and then everybody dies. Still, like, there's a famous, like, scene that you would see in like a lot of these movies, you know, Hunt for Red October, that kind of thing, where you got to launch the nukes, right? And we don't just give nukes to one person, I guess, except the president. But like the idea is, do you really want nukes to be like any ship captain could just wake up one day and go, woohoo, I've gone crazy, I'm going to launch me some nukes? That doesn't sound like a very safe idea. You know what happens? No. No. So it takes two people. So the captain of like the nuclear submarine or whatever gets orders, and he has to verify, yes, we have valid orders. We are going to destroy the world and launch our nukes, right? And he has a key. Technically, it's two keys, but they're the same key. And he has to go over here and put his key in. And then the first mate or the second in command, they get the orders as well. They also have to verify that, yeah, okay, these look like legit orders. I agree. Time to launch the nukes. And he has a different key. It goes over here. And crucially, the two places are far enough apart that you cannot turn keys in both of them at the same time. And they have to both turn the keys at the same time in order to launch the nukes. This way, like, you could envision one guy going, like, you could envision the captain going crazy and deciding to launch some nukes because he's part of some weird death cult or something, right? But it's pretty unlikely that his first mate is going to go, yeah, that seems like a good idea. Why don't we just randomly launch nukes at people? And so having this second confirmation makes it a lot safer. How is this relevant to the immune system? Antibodies are kind of the nukes of your immune system. They're ridiculously powerful. Pretty much anything that gets labeled by an antibody gets destroyed. Would you want all of that power to be held in the hands of just like some schmucky B cell that you've got, you know, billions of in your body, and maybe it didn't get like trained properly, or maybe it got a mutation so that now it recognizes your body and just go, I'm going to make antibodies against the pancreas. Here, have some diabetes. That sounds like a bad idea. So just like here, your B cells normally require two activations. One, they have to decide for themselves, yeah, I should make some antibodies. And two, they have to get confirmation or permission from another cell that has reached that conclusion independently. How's that happen? Well, let's start here. We've got our B cells floating around again, and I got bored of using bacteria, so I used a virus this time. All right, so we got a virus coming in. The virus, you know, it's floating around in the blood or in the lymph or the wherever. It's bumping into lots of B cells just randomly on accident. Bumps into this guy, nothing happens, it doesn't stick. Bumps into this guy, nothing happens, doesn't stick. Bumps into this guy, and this B cell is like, I know how to grab onto this. My antibody, my B cell receptor matches this antigen perfectly. I can grab it. And it goes, I have reached my purpose in life. But it's not going to activate yet. What is it going to do? It's going to take whatever it was that it bound inside. And sometimes it takes the whole thing inside if it's small enough. Sometimes it just yoinks off the whatever antigen it bound to and just takes the antigen inside. But either way, whatever it bound to, it's going to take that thing inside. And then, as you noted earlier, B cells are also antigen-presenting cells. Now, their purpose is a little bit different than the phagocytes. They're really more here to receive information than to send it. But the mechanism is the same. So we're now going to display this antigen that we just bound to on an MHC2. And again, we're not going to do any good. We're not going to get confirmation just floating around here. We need to go where there's a lot of things that can confirm for us, that can give us permission to make antibodies. And that means we need some T cells, hence T-dependent. Now, I said this story has multiple parts. We're going to let our partially activated B cell here. We're going to let him aside for a little moment, and we're going to return to our story of the antigen-presenting cell. Now, our poor antigen-presenting cell, our poor dendritic cell, has been hanging out inside the lymph node. Yes. For a little bit. And the reason why it went to the lymph node is where you're going to find lots of lymphocytes. And what this guy needs to do is he needs to report his information to the appropriate lymphocyte, which is going to, in this case, be a helper T lymphocyte. Just like I said, every B cell has its own unique independent B cell receptor. Every T cell has its own unique independent T cell receptor. T cell receptors share some similarities with B cell receptors, but they're not exactly the same. Like, first off, they aren't shaped like antibodies, but they do still have a constant region and a variable region. And in this case, a large chunk of the constant region, what it does is it binds to MHCs. If you're on a helper T cell, it's going to bind specifically to MHC2. And then attached to that is a variable region that binds a thing, right? And every single one of these receptors is going to bind a different thing randomly. And you'll have billions of these guys in your body. So they can't just see anything. They can't see things that are just floating around in your blood. Like a B cell, if a B cell has a receptor that fits this, say this here, and this is like just floating around, it can go, aha, yoink, I bind to this, and I take it inside. But if you have a T cell receptor, it can't bind this because it's not being displayed on an MHC2. Now, go ahead and grab this by the end and display it to me. Right? And now, if I am a helper T cell, I can go, ah, I see you. Yep, you're displaying this to me on the right thing. Now I can grab it. So it can only see things that are being displayed on an MHC2, which means it only sees foreign antigens, because only foreign antigens get displayed on an MHC2. So here we have our dendritic cell. It's gonna go through a lot more than two. You can kind of think of the lymph node as like Tinder for your immune system. It exists so that you can go through a lot of false matches really, really fast before you find the thing that you match with, right? So it's like, no. Ah, that's a good match. Because there's like tons and tons of helper T cells in here, and this can meet up with a whole lot of them. It's a speed dating thing. And it goes up here, right, match. And this helper T cell can read that, and it's being displayed on an MHC2, so it can bind. And this helper T cell becomes active. And it's safe to activate because it does not make antibodies, not by itself. So when this helper T cell becomes active, it says, ah, dendritic cell, I see that you have displayed for me this horrible antigen from this nasty virus that I must get rid of, or that I must help in getting rid of because I'm a helper. I shall take this information far and wide, all throughout the body. I will assemble our forces. I will find somebody who knows how to fight this horrible virus, and together we will defeat it. Doesn't actually say that, but yeah. Yes. Right. So the T cell is, now that it's active, it's going to like replicate a whole bunch. It's going to make a whole bunch of copies of itself, and they're going to go from this lymph node out to all the rest of the body. Okay, what happens next? Well, in any good story, now we're going to have our two plot lines come together. Our two main characters are going to meet up, join forces, and destroy the evil virus invading the body. So here we have the B cell and antigen-presenting cell stories unite. So here we have our activated T cell. It's moved off. You saw it leave. It went off to a different, yeah?
When you're saying activated T cell, is this the helper T cell?
Helper T cell, yes. So that the TH that you see there, yeah, that means helper.
Okay.
We're not talking about cytotoxic yet. We'll get to that next week. So here we have an activated helper T cell or CD4 T cell, and it has gone to this lymph node. And this is the lymph node that our friend, the B cell, shows up in. And this B cell, it's not fully active yet. It's still what we call naive. It hasn't been fully activated because it's still looking for that second key. And it's gone to the lymph node. And what it's going to do is say, I found this thing, and I think I should make antibodies against it. Can anyone confirm for me that I'm not crazy and I should just go ahead and do this? And it's going to cycle around all of the various helper T cells that are around here and look for something that binds it. Now, remember, what's that being displayed on? An MHC2. Remember, B cells are antigen-presenting cells. So can this helper T cell receptor, can it bind to it? Yes, because it's being displayed on an MHC2, and it matches the binding pocket. So when these two cells meet up, that helper T cell says, I see you know how to recognize this virus. I carry confirmation from a very noble dendritic cell that has brought to me saying that he also found this damn thing floating around, and you have permission to destroy it. And so you saw little stars that came out there? Right. So it releases cytokines, usually a type of interleukin, right? And it releases these interleukins, and the B cell is now going to be fully activated by those interleukins. And what does it do when it becomes active? Well, a couple of different things. First off, it's going to start multiplying. So it generates a whole lot more B cells, copies of itself. All of these copies of it are going to make the same antibody, or make the same B cell receptor. Now, next thing it's going to do is develop into plasma cells. Remember what I said plasma cells do? They make antibodies. The helper T cell doesn't go away. It's going to continue sitting around helping. And the way it's going to help is secreting more cytokines that are going to help coordinate and control the B cells. So one of the first things that it does is it's going to make some cytokines that are going to turn some of the active B cells into memory B cells. Memory B cells are similar to normal B cells, except they're very, very long-lived. And there are going to be a whole lot more of them, and they're going to be easier to activate. So that next time you encounter this thing, instead of there being like one B cell in your body that can recognize it, you've got a few thousand. And so therefore, they're going to find their target much faster. All right, so you're going to get a few memory B cells. Cool, they're going to go remember things. Now, all of these plasma cells, they make antibodies. Specifically, the antibody that they make, the antibody all plasma cells make to start is IgM. Stands for interglobulin M. Or, immunoglobulin M. An immunoglobulin is it's the class of protein that an antibody is. And I've done it like that because IgM is a pentamer. It's actually 5 antibodies glued together.
So you said that's the first one that they produce?
This is the first one that they produce. All of them start off producing this. And IgM is good at doing some things, but is not for everything. So another thing that the active helper T cell does is it produces cytokines that are going to cause class switching. And so this plasma cell was exposed to it, and now it's making IgG. IgG is a monomer antibody. It's actually the workhorse. It's the one that's made most abundantly and the one that's responsible for most immunity? IgG. Oh, by the way, when these memory B cells are going to activate later on, if you're exposed again, they're going to start making IgG right away. At least some of them are. Here, we're going to take this one, and it's going to class switch to make IgA, which is a dimer. This is actually found in mucus and is the most abundant antibody in the body and helps to protect you from getting infected through your mucous membranes. And like I said, I'm going to upload a video about antibodies that's going to go through all the different classes and exactly what they do. So, without this helper T cell, what do you have?
IgN.
You have IgM. All you have is the default plasma cell, which makes IgM. Do you have any memory? No. Do you have any IgG? No. Do you remember the T-independent method? The one key method? Remember what I said are two things it doesn't have? It doesn't have class switching. It only makes IgM. And it doesn't have memory. Because you require the helper T cell for those two things to develop. And without that helper T cell, all you're stuck with is your base level plasma cell. And even then, only against a very limited variety of antigens. Anyone have any questions about this? Yeah. How memory B cells what? Oh, it's the helper T cells going to release a cytokine. You don't need to know what cytokine. It's one of the interleukins. And that causes the B cell to differentiate. Remember we talked about differentiation? To differentiate into a memory B cell. And some antigens and some helper T cells are going to produce more of a memory and some are going to produce less. We don't exactly know why. Yes?
So if I can remember right from homework, the IgG is the only antibody that can cross the placenta.
That's correct.
So in the case of, does that help in the case of like a female being pregnant?
Well, yes, it's the only it's the only antibody that crosses the placental membrane. Actually, because IgG is actually fairly long lasting, it lasts for several months in the body. So when a baby is, and we'll talk about this next week, probably next week. When a baby is born, will it have antibodies in its blood?
IgG.
It'll have IgG from its mother because it's born without an immune system. So the IgG is what's going to protect it for the first few months. But good question, yeah. Relatively little infection happens in the womb, but right as you're coming out, a lot more. So this is basically just an overview of what I just told you. And just to show you the power of memory, right? Here you see a person exposed to an antigen for the first time. It takes a long time for this two-key method, the T-dependent method, to happen. Because you've got to have, like, the right B cell runs into the right thing. And an antigen-presenting cell has to run into that same thing, eat it, find exactly the right T cell, and then that T cell has to find that first B cell again. All of that takes time. It can take anywhere from, like, days to weeks, maybe months sometimes. So here we have a pretty average case. At first exposure, you're not making any antibodies at all until 10 days. You don't make peak IgM until about like almost 15 days. Remember, IgG is really the workhorse. You're not making peak IgG until 20 days. That's when you're going to really start destroying this infection. That's from first exposure, not from first symptoms. Here is the second time you're exposed. You see, you're going to start making IgM like way faster at about 4 days, or IgM. And you're also going to start making IgG at the same time. There's no lag there. So you reach peak IgM within a few days, so like at the 7-day mark. And IgG is also going to come up much faster. And you're just going to make a whole lot more of it, and you're going to make it for a longer period of time. That's the power of memory. Yeah.
So in this case, would a secondary antigen exposure trigger like you to feel worse? Like if you get the exact same flu 2 times, so primary, secondary, the secondary one you're going to feel like more trash because more antibodies, a stronger immune response?
No, because antibodies aren't generally what makes you feel like trash. Mostly it's inflammation. Inflammation is what's keeping you alive till here. So now, usually also you're going to have, because you're just, so the number of bacteria, the number of viruses you have in your body grows exponentially. Remember the growth curve? Yeah. So with that, on re-exposure, you're going to squash the infection flat before, like, in half the amount of time. And half the amount of time doesn't mean there's half as much. It means there's like 1% as much of it there. So it's going to have a lot less time to do damage to you as well, which is the other thing that makes you feel like crap. Yeah.
Vaccinations would be creating the memory, so we go into secondary.
Vaccinations are doing this without it killing you. And we're going to have a whole thing on vaccinations. We're going to have a whole day on it. All right. Any other questions? Cool. That was interestingly experimental in a way that didn't quite work out. I'm going to have to work on that.